Today we'll talk about eukaryotic RNA processing, finish with it, and then about translation. So we said that in eukaryotes, when protein coding genes are being expressed, there is promoter recognition, of course, transcription initiation, and chain elongation. And that primary transcript that is created by the process of transcription needs to be processed, because that primary transcript is far from what it will look like in its mature form as an mRNA, as a mature mRNA. We mentioned kind of briefly, they're grayed out now, but we mentioned 5' capping and 3' polyadenylation to post-transcriptional modifications of that primary transcript that are needed for protection of that mRNA molecule so that the cell doesn't mistake it for a foreign RNA, particularly a virus. And also for initiating translation, because the 5' cap and the poly A in the 3' end of that molecule basically tell the cell this is a whole mRNA sequence. We're not missing anything. So the one that we didn't get a chance to discuss yet is splicing, which is a process that applies to eukaryotic genes, except those that we call intron-less, for reasons that will become apparent in a second. So splicing as a process is, and many of you may have heard of these before in previous courses, but a refresher, as a process is a mechanism by which the nucleus, let's say the cell, but in the nucleus, the cell takes that primary transcript that is much longer than what the mature mRNA will be, and it takes out a series of chunks of sequence from within that RNA molecule, that pre-mRNA we call it, and stitches together the remaining ones. So just an important definition, simple but important definition, any sequence that was in the primary transcript and ends up in the mature mRNA, and is present in the mature mRNA, we call exons. Anything that got taken out are called introns. So see in this diagram, these green boxes here are the introns because they're not present in the mature mRNA. Yeah?
So are introns, because they're not needed, are the only reason they're there is because that's how it was coded from the DNA that is just...
That's right. The introns are present in the DNA sequence that was transcribed into that primary transcript.
But it's just kind of useless in the grand scheme.
If they were totally useless, you would think that evolution would have gotten rid of them. So there's a bunch of theories that say that there are uses for them, and in fact, we'll discuss a couple of them in about 3 lectures time when we talk about gene expression regulation and alternative splicing. So there's already some evolutionary value or adaptive value in having introns, because then cells can shuffle combinations of exons and give rise to different mature mRNAs. There are other theories that say that introns may have served other evolutionary advantages or purposes. Saying purpose in biology is a little loaded, but that they may have other adaptations. The point is that from the point of view of protein translation, yes, introns, you could think of them as useless sequences that need to be taken out. So let's look into that process in a bit more detail. If you were to look at any intron, the sequence that needs to be taken out, you would see that on its 5' end, on what we call the exon-intron junction, there's going to be a sequence that is not just any sequence. It's a consensus sequence that, unlike those that we saw before that were very firm, TAA, TAA, or the -35, they're not that strict, but they're still consensus sequences. These ends here are there to represent a sequence of nucleotides that won't be just any sequence, just any combination. They always have some sort of they fall within a limited set of options, of possibilities, right? Now, the one thing that is invariant about these consensus sequences that are at the boundary between an exon and the downstream intron is that GU on the intron side, right? And it is a U because, mind you, once again, This is an RNA molecule. We're off DNA already. This is the primary RNA transcript, and now we're working on an RNA molecule, so GU. On the other end of the intron, in the intron exon boundary, but downstream one, same story, except that consensus sequence starts with an AG that is invariant. There's always an AG. And then a bunch of sequences that, again, not a very firm consensus sequence, but always falling within a rather limited set of options. And then somewhere in the middle, there is what we call the branch point. Again, a loose consensus sequence with a critical A somewhere in the middle. So I point all this out because there is nothing in the RNA molecule from a chemical point of view that will distinguish the exon-intron boundaries from the rest of the RNA molecule. It's not like at the boundary, I don't know, there is a triphosphotriester bond or a diphosphodiester bond or anything like that. There is no chemical modifications of those nucleotides. There is nothing weird going on. It's all by sequence that a cell and the machinery that does splicing within the cell will know that there is exon-intron boundary there, an intron-exon boundary here, and a branch point here. We give these names, things names. So the 5' splice site, that upstream exon-intron boundary, we call it the donor site, and the 3' splice site we call the acceptor site. And then the rest, I think I said. So the way this works from a molecular perspective is there is a machine in cells called the spliceosome. The spliceosome is a combination of proteins and very special non-coding RNA sequences. One of the many non-coding RNA sequences present in cells that we call small nucleolar RNAs, or small nuclear RNAs, snRNAs. The job of those snRNAs is to recognize these donor and acceptor sequences on both ends of that intron through complementary base pairing, right? It doesn't have to be a perfect 100% match to those donor and acceptor sites, but there's going to be some degree of complementarity, and that is how the spliceosome knows where the donor sites and the acceptor sites are. So splicing begins when the spliceosome recognizes a donor site and an acceptor site. And then here on the left, I'm just showing the beginning of the process and the very end of the process. And here on the right, I'm showing it stepwise. So what happens is there is that recognition of the donor and the acceptor site. Some of the proteins within the spliceosome have enzymatic activity, so they will cut at the donor site here. And they will catalyze a new chemical reaction that creates a covalent bond between the UG and that critical A within the branch point. So now there is an actual covalent bond illustrated there by that GPA, meaning a G bonded to A through a regular phosphodiester bond, forming what we call in technical jargon a lariat. The same spliceosome will then produce another cut at the acceptor site, and enzymes within that spliceosome will then splice together those two exons. Yes?
Is the 5' site always going to have GU and the 3' to have AG?
Always. So the question is, will the 5' have a GU and the 3' end of that intron have an AG? Always. Now, it's not only those nucleotides that matter, it's also some of the preceding sequence and some of the post-seeding sequence. It's not like the spliceosome will recognize as a donor site any GU and any AG in a primary transcript. They have to be at the end of some loose consensus sequence and at the beginning of some loose consensus sequence. Yeah? Yes?
So why doesn't it just cut it in the center of it? Why does it have to cut it at the donor site exactly?
If it cut at the center of it, then if it spliced those ends together again, it would reconstitute the primary transcript as it was, right? So in order to take something out from this primary transcript, you need two cuts. Take that thing in the middle out, and then rejoin the ends that were produced by those two cuts. Does that make sense? Splicing, I know that nowadays it sounds a little, like ancient history, but splicing was something that movie editors would do. They would actually cut the strip of film and then splice the two resulting ends together, and that is how they would cut out certain scenes from a movie, right? So this is the same thing. We're splicing out, we say, the intron, but what we're really doing is cutting out the intron and splicing together the two exons. Yeah? This lariat here is not a common structure for an RNA molecule, right? RNA molecules can form hairpins, yes, they can form stretches of secondary structure, but this whole business of looping around themselves and forming an intramolecular covalent bond is something that is not very normal. And the cell recognizes it as an intron lariat and destroys it. And when it destroys it, it will break those lariats down to all of its nucleotide components, and those nucleotides will be recycled to be used as building blocks for the next RNA molecule that gets transcribed from DNA. Yeah? Questions so far? Yes. The question is, when it forms the lariat, the reason I'm repeating the question is you're far away, so I want to make sure that it's recording in the lecture capture. So the question was, was I saying that this lariat gets destroyed by the cell? Yes, that's exactly what I'm saying. So the intron gets spliced out, forms this lariat, and the chemical structure of that lariat is quickly recognized by special enzymes within the cell that will know to destroy this. Because this thing has no use otherwise, and it would be a waste of resources, right? So it recycles the building blocks, the Lego pieces. Okay, so a very important concept here, once again, is the spliceosome sees donor sites and acceptor sites and a branch site somewhere in the middle, a branch point somewhere in the middle, right? But what happens is that sometimes the spliceosome may miss certain donor sites or certain acceptor sites. It may not see them. We will explore this in a bit more detail in a couple of weeks when we talk about gene expression regulation. But the bottom line is that the same primary transcript that is shown twice here can be spliced differently through two different splicing events, right? So the one on top, every donor site was recognized and every acceptor site was recognized. Sorry, I pointed to the wrong acceptor site. And what happens is all of the introns from these primary transcript were spliced out and all the exons were spliced together. But in the example here at the bottom, what happened was that this particular acceptor site was missed. Somehow the spliceosome didn't see it. And it used instead the next acceptor site. What that effectively does is it joins exon 1 with exon 3. And it skips exon 2. This is, again, the result of the fact that there is nothing in an intron sequence other than those donor and acceptor sites that tell the spliceosome this is the intron. From here to here is that you should cut. No, the spliceosome doesn't know any better. It knows to recognize donor sites and acceptor sites, and sometimes it misses a few of them in between, and it will skip exons, whole exons altogether. And what's going to happen in this case, and we'll see this a little later, is that now this lariat that forms will include somewhere that exon 2. So that exon 2 is destroyed along with the lariat, gone. And that particular mature mRNA that came from these splicing event doesn't have exon 2. It goes from 1 to 3. Yes?
Would it be accurate to think that the introns stay in the cell and the exons go out of the cell?
Go out of the cell? The introns? And the
The introns move out of the cell.
move out of, move out of the cell. The introns move out of the cell and the exons stay inside of the cell.
Introns stay in the exon rest of the cell. Like to be translated.
The nucleus, you mean, not the cell. The nucleus. So the question here is it accurate to think that exons exit the nucleus whereas introns stay in the nucleus? Sure. Exons are the ones that effectively will exit the nucleus and be translated at some point. Though we'll have to qualify that statement a little bit in a second. And introns stay in the nucleus and are destroyed in the nucleus. The only way that your statement might be a little misleading or inaccurate is this idea that the introns hang out in the nucleus forever, when in reality they're just destroyed. So it's probably more accurate to say exons stay, introns are gone. All right, so that's splicing in a nutshell. And the very last step in the process of RNA processing in eukaryotes, it's exporting those mRNAs from the nucleus to the cytoplasm. We're not going to go into any of that in detail, but let me tell you something that should be that hard to imagine, that this is also a carefully regulated process. The nucleus doesn't want to export any half-processed mRNAs, right? It wants to export mRNAs that have been properly spliced, properly polyadenylated, and properly 5' capped, right? So there are a series of checkpoints and control mechanisms that ensure that that nuclear export is for fully formed mature mRNAs. Okay. So after all is said and done, and the mRNA has been fully processed in eukaryotes, it's time for translation. And now we come again back into the middle, in between bacteria and eukaryotes, because aside from a few molecular details, the processes are very, very similar in both of them, right? They both occur in the cytoplasm, right? Because the inside of a bacterium is the cytoplasm, and the cytoplasm of eukaryotic cells, and both use similar machinery. So let's talk about translation very broadly as a process, and let me address one of the most common and sad, I guess, misconceptions, and we kind of touched on this already. Very common, so don't feel bad if you thought this way. It's a very common mistake, and that is to think that the AUG that signals the start of translation is at the very beginning of an mRNA molecule. That the first 3 letters of a mature mRNA are AUG, because that's the start code. We talked about this from the point of view of transcription. There were some discussions here in which it kind of got thrown around that the TSS is the AUG. It's not. The TSS, any nucleotide, could be anything, right? The AUG is what starts or signals the start of translation, and it is what the ribosome uses to say, okay, from here on out, I'm going to start translating. So conceptually, not much different from a TSS, but it's the translation start site. But we simply call it the start codon. I don't want to confuse you anymore. And then there is another very special codon that we call the stop codon. There's actually 3 of them, right? So they're indicated here in this version of the codis, of the genetic codis. There's so many out there. You can find them in circle form, in the boxes, like all kinds of formats. So translation is the process by which, from that start codon, that start AUG, to the very last stop codon, groups of nucleotides, 3 nucleotides that we call codons, get translated into 1 amino acid. So the ribosome reads 3 letters at a time and knows what amino acid corresponds to those letters. We humans created these tables, but this is essentially what we learned ribosomes do inside of cells. Ribosomes don't have these tables. They know it naturally. We figured out that that's what they do. Okay? So let's talk about the hardware a little bit. Translation happens in ribosomes. Ribosomes are massive macromolecular complexes that are mixtures of special RNAs that we call ribosomal RNAs. Here are some of them shown. So there's more than one ribosomal RNA forming part of ribosomes, both in prokaryotes and eukaryotes. They differ in size and in sequence, obviously, but they're analogous in every other respect. And that's for a reason, because they were, inherited from their common ancestor. And one thing about ribosomal RNAs is that they get transcribed in one of these polycistronic RNAs, these RNA transcripts that are much longer than any one of these final ribosomal RNA molecules. So they get transcribed together as part of a huge polycistronic mRNA, and that RNA gets chopped up into pieces, and some of them will form these ribosomal RNA structures, and whatever sequence there was in between them will get degraded, destroyed, recycled, the usual. And these ribosomal RNAs serve as a scaffold for a bunch of ribosomal proteins to combine with them and form the ribosomes. And as we will see in a couple of slides, ribosomes are formed by two subunits. There is the small ribosomal subunit and the large ribosomal subunit, both of which have a combination of ribosomal RNAs and ribosomal proteins. Questions, comments? All right. What's the other piece of hardware in translation? It's the transfer RNAs. Transfer RNAs are the ones that bring amino acids to the ribosome so the ribosome can stitch them together into a protein molecule. And they are also the ones that can literally read those codons in the messenger RNA. It is common to say that the ribosome reads the mRNA and translates it into a protein. The one actually doing the reading is the transfer RNAs. They are the ones that base pair with each of those codons in the messenger RNA. So they are the ones, in a way, that have the ability to know what combination of 3 nucleotides are in each codon within that mRNA. What the ribosome provides is a physical structure for the transfer RNAs to find those codons in the mRNA, and the enzymatic activity that allows for these amino acids that are brought by the transfer RNAs to be stitched together into a polypeptide sequence. You'll hear me now use protein and polypeptide kind of interchangeably. There's a reason for that. When these chains of amino acids are being stitched together in a ribosome, kind of unfair to call them a protein yet. Many of them are not really fully folded and functional. So when they're being produced at the ribosome, for now, they're just a chain of peptides or chain of amino acids bound by peptidic bonds. So we call them polypeptides. Sometimes you'll hear me say protein being produced, sometimes the polypeptide being produced. They're synonyms in this context. Okay? All right, so these transfer RNAs have two key components to them. They all look very similar. They're all produced also by longer polycistronic mRNAs that get polycistronic RNAs that get chopped up and give rise to several transfer RNAs simultaneously. They all have a similar structure. They all have these sort of L-shaped structure when they fold in space, and they fold via all of these intramolecular stretches of complementary and anti-parallel base pairing that we mentioned before. And they end up with a shape that exposes two ends. There is the anticodon end, where there's going to be 3 key nucleotides that are the ones reading the mRNA and doing the actual base pairing with the corresponding codons in the mRNA molecule. So these 3 nucleotides in the anticodon sequence of the transfer RNA are the ones that are actually reading the message in the messenger RNA. And then on the other end, there is the the aminoacyl site, and that means the area of these tRNA molecules where the amino acids are covalently bound until they're transferred by ribosomal proteins from this tRNA molecule to the growing polypeptide chain. Okay? Like I said for transcription, I'm going to emphasize this once again. We'll go through the process via these static images, but I strongly encourage you, even if you're not the most visual of learners, I strongly encourage you to look at the hundreds of videos, educational videos about translation that are out there, because they will present this process as a movie, as a very dynamic process, and that goes a long way, okay? But here in class, we'll talk about all of these with still images. So before we go actually into translation, I just wanted to address a point that sometimes generates a little confusion. So we keep saying each tRNA molecule pairs up the right anticodon to the right amino acid on the other end. If they didn't do that, then the anticodon will read the same codon in mRNAs every time, but it will bring a different amino acid every time. That's not very useful for the cell, right? The cell needs that every time a given codon is present in mRNA, the transfer RNA bringing an amino acid brings the same amino acid, the amino acid that corresponds to that codon, right? So how does these tRNAs know which amino acid they should bring? Well, that's the job of these very special enzymes. They're a mouthful, but these enzymes are called, come on, marker, there you go. These enzymes here are called tRNA aminoacyl transferases. And it's in the name, right? So what they do is they transfer an amino acid through again, an ester bond to the tRNA. And how do they know which amino acid to load into the tRNA, or as we would say in jargon, what amino acid to charge onto that tRNA? It's all, again, this is an enzyme, right? So enzymes have this ability to recognize their substrates in a very biochemically exquisite manner. So these tRNA aminoacyl transferases will have a region in them that will base pair, or not base pair, but interact with the anticodon in such a manner that they know exactly what anticodon they're dealing with. And then somewhere else, there's going to be another binding pocket or another catalytic site that will know to bring phenylalanine, in this example, or whatever the amino acid is, to these tRNA. So the specificity in the pairing of the amino acid to the corresponding tRNA is given by these enzymes. And if you guys remember your enzymatic days and biochemistry days, enzymes are super specific for the substrates. They won't react or interact or work with just anything. So these tRNA aminoacyl transferases are not different. They have binding sites for the right anticodon and binding sites for the right amino acid, such that the only amino acid that they can charge a tRNA with is the one that corresponds to that anticodon. This charging reaction is a very energetically costly reaction. So enzymes need a little help, and what they do is they hydrolyze in the process of charging that tRNA, they hydrolyze an ATP molecule, and they use the energy that comes from that ATP hydrolysis to charge the tRNA with its corresponding amino acid. Okay. Questions, anyone? Yeah. No, what happens here is so it releases 2 phosphates. So this ATP has 3 phosphates, and what it does is it breaks that triphosphate between phosphate 1 and 2, so it will leave 1 phosphate linked to the A, so you end up with an A monophosphate and then a diphosphate that gets released. Yeah?
So basically, it just makes a pyrophosphate and build it on AMP.
AMP, yeah.
And then the AMP gets down to the phenolphthalein.
No, that's release. So all the enzyme is doing is capturing the energy from that hydrolysis. It's coupling a highly exergonic reaction, that is the hydrolysis of ATP, with an endergonic reaction that is charging a tRNA with an amino acid, something that it's energetically costly. Okay, let's move along. So now we're going to go through the process, and we'll point out differences where there exist. So you have your mature mRNA, and what happens first is there are these small ribosomal subunits that are preloaded with the tRNA for AUG. Because, as I said a couple times now, AUG is the start codon. And it is the start codon in bacteria, it's the start codon in eukaryotes, meaning animals, fungi, plants, right? Archaea. So if all proteins or all polypeptides are going to start at that AUG, you need the tRNA that recognizes that AUG. So I guess life or evolution figured this out, and what evolution did was to just preload the small ribosomal subunit with that tRNA for AUG. And now these things are patrolling the cytoplasm looking for mRNAs to translate. When they find an mRNA, in the case of prokaryotes, they will recognize the 5' end of an mRNA through binding to a sequence that we call the Shine-Dalgarno sequence. The Shine-Dalgarno is almost like a signal in bacteria that says, hey, here's the beginning of an mRNA. Why don't you come bind this mRNA molecule here, and then you go from here. In eukaryotes, that signal is the 5' cap. The 5' cap doesn't have a sequence per se, but it is chemically distinct enough that can be recognized by that small ribosomal subunit preloaded with the tRNA for AUG, such that it can bind to it, and it knows, quote unquote, that it bound the mRNA somewhere at the beginning of the molecule. And once these binding happen, then the complex, and when I say complex, I mean the small ribosomal subunit with preloaded tRNA, will start scanning that mRNA molecule looking for that AUG. So when the small subunit plus the tRNA for, by the way, I keep saying the tRNA for AUG, the corresponding amino acid is methionine. So when the small subunit plus the tRNA for methionine and a bunch of other proteins that are accompanying these two find that AUG, what happens is they will stop, they will, quote unquote, detect that they found the AUG, so they will undergo a very special conformational change that now allows for the assembly of the rest of the ribosome, the large ribosomal subunit. So when the large ribosomal subunit binds the small and they form a whole ribosome, what the large ribosomal subunit brings with it is 3 sites in it. There is the A, the P, and the E site. When the large ribosomal subunit lands on the AUG and the small ribosomal subunit, that AUG and the tRNA for methionine end up in the P site. Okay? That's how the large subunit lands, leaving the AUG and the tRNA for methionine in the P site. And now, the A site offers the space for the next tRNA to come in and bring the corresponding amino acid. So we can think of it this way. That A site is exposed to the world, meaning the cytoplasm. And in that cytoplasm, there's a whole bunch of tRNAs that are floating around and bouncing around, and they might come and see that A site and they go in. But if there is no good match between their anticodon and the corresponding codon in the mRNA, they won't stay. They will bounce right off. Right? that A site is not the most comfortable place for tRNAs unless their anticodon matches perfectly the codon in the mRNA. And when I say matching, I mean base pairing the old-fashioned way, right? So you have the codon, 3 letters in the 5' to 3' orientation, and they have to be base paired by 3 letters that are in the opposite orientation, 3' to 5'. I said 3' to 5', and of course, following the rules of complementarity, A to U and C to G. So if the right tRNA lands on the A site and base pairs with that codon, it can stay in that site long enough that the ribosome recognizes that that A site has been occupied, and it will catalyze the transfer of the first amino acid, methionine, to the second, in this particular example, phenylalanine. Okay? So the methionine is covalently bound or transferred to the phenylalanine to form a methionine-phenylalanine dipeptide. Now we have 2 amino acids bound by a peptidic bond. We call that a dipeptide. More generally, the methionine is covalently bound transferred to whatever that second amino acid is. Sometimes these diagrams, in spirit of showing you an example, will tell you that methionine is transferred to phenylalanine, and next thing you know is most students will think that the second amino acid in every protein is phenylalanine. It's not. The first one is always methionine, but the second one could be anything. It depends on the codon that was present in that mRNA. Now, the ribosome senses, and when I say senses, it's not like they have eyes and ears or noses or anything, but chemically they can detect that the tRNA at the P site is empty now, because when that tRNA gives up its amino acid, it will change in its 3D shape. So the ribosome detects that the tRNA at the P site is empty, and that triggers what we call a translation. So the whole ribosome moves down 1 position, 1 codon downstream, right? Now imagine that the tRNAs are not interacting with the ribosome. They occupy spaces in the ribosome, but they're interacting with the mRNA. So when the whole ribosome moves, the tRNAs stay where they were. The ribosome moves, and now what used to be at the P site moved to the E site. So that empty tRNA that used to be at the P site now kind of ended up, because the ribosome moved, ended up in the E site. And E stands for exit. So now this empty tRNA can exit the ribosome. The translation of the ribosome also moved the dipeptide to the P site and created a new A site that is empty. So now we repeat the process, right? We have a transfer at the P site with 2 amino acids bound to it now, and an empty A. That creates the space for these tRNAs to start coming into the A site and just trying it out, see if they fit. Most of them will bounce off, but one of them, the one that has the anticodon that corresponds to the next codon in that sequence, will bind and stay. And when that happens, the whole process repeats. So the next tRNA brings arginine. The ribosome senses that the A site has been now occupied by the right tRNA with the right amino acid in it, and there will be a transfer of the dipeptide to the 3rd amino acid to form a tripeptide. And again, in this particular example, we'll have a methionine, phenylalanine, arginine tripeptide, but more generally speaking, it'll be methionine to amino acid 2 to amino acid 3.
So I just want to make sure. So the very first tRNA that goes to attach to the ribosome goes to the E site, and then the next one goes to the A site, which then causes it to shift afterwards. And what was once that.
Think about it. So these A, P, and E sites are like windows, right, in a wall, and the wall is moving, right? So the mRNA has all these transfers bound to it, and the ribosome with the walls, or with the wall, with the windows moves. So what you used to see through the E window was nothing, empty. Now the wall moved, and now through the E window, I see this empty tRNA. What used to be in the P window now moved to the A, right? So, what? This is why you need to be. Yeah, this is why you need to watch the videos. I was like, I have to go into full-blown theatrics, and that's not good for my reputation out there. So when the ribosome is moving to our right, right? So when the ribosome moves, whatever used to be at the P, that you could see through the P window, now you can see through the E window. And whatever you could see through the A window, now you can see through the P window. And whatever you could see through the A window is empty now. Does that make sense now?
Makes sense.
Yeah? Yes?
On the anti-tRNA that's on the exit site, does it immediately exit the ribosome when it moves to that site, or does it wait for like the other?
There may be an answer to that, I don't know it. This cartoon makes it look like he waits, but it's a cartoon, so I don't know. Good question, I don't have an answer. Okay. So this process continues over and over and over with the ribosome moving downstream on that mRNA. This process repeats and repeats, one codon at a time. And before we get to the stop codon and what happens then, I'd like to ask you a question. So this transfer of energy, or transfer of amino acids, and the sliding of the ribosome requires energy. Where do you think that energy is coming from? It's not ATP, it's its cousin, GTP. So ribosome translation actually consumes, there are two stages at which this consumption of GTP, not ATP, GTP. Now, one thing that I'd like to emphasize before we get to the end of translation is, remember transcription? I asked a similar question. I asked, where is the energy to bind the next nucleotide, the incoming nucleotide, to that growing chain of RNA, right? And the answer was, oh, it's in the molecule itself. So if an A was to be added to the RNA molecule, what was coming in was an ATP, and RNA polymerase was using the energy stored in ATP to carry out that reaction. If the next nucleotide that was coming was a C, it came in the form of a CTP, and the RNA polymerase would hydrolyze that CTP and extract the energy that it needed. Here, it is super tempting to think that the energy needed to carry out this transfer of an amino acid to the next, and then from that dipeptide to the next to form a tripeptide and so forth, comes from the high energy bond that stays between the tRNA and the amino acid. It is empty because that's a phosphodiester bond, of sorts that forms with the tRNA. So it is tempting to think, oh, the energy for the peptide transfer is there. Well, no, most of the energy was spent in charging the tRNA. That tRNA charged with an amino acid is not that energetically, it doesn't have enough energy to move this process along. So the ribosome needs a little help, and it will use actually two GTP molecules. So the energy is provided by the hydrolysis of two GTP molecules by a group of elongation factors that come to replace the IFs. I never really explained what IFs are. IFs are initiation factors, and these are proteins that kind of hang out with the small ribosomal subunit and that preloaded tRNA for methionine and are with them while they're floating around in the cytoplasm looking for mRNAs. When they bind that mRNA at either the Shine-Dalgarno in bacteria or the 5' cap in eukaryotes, and when they're scanning for that AUG, but when they find the AUG and the whole ribosome forms, these initiation factors leave and elongation factors come in that are the ones responsible for this GTP hydrolysis business and to move the process along. Okay? All right, so let's talk about the end of this process. When ribosome moves until it finds, at some point, it will bump into a stop codon. When the stop codon is in the A site, there is no tRNA for stop codons. There is no tRNA that has an anticodon that base pairs with stop codons. So all the tRNAs will come in and bounce off, come in and bounce off. There's no tRNA that can stay there. The only thing that can come into an A site with a stop codon and stay is a protein that we call release factor. And the release factor kind of tricks the ribosome because the ribosome thinks, oh, my A site has been occupied now. I can transfer the polypeptide that I've been building all this time to the new amino acid. But it turns out that that release factor is not offering an amino acid for the peptide to be transferred to. So what does the ribosome do? It just lets go of that peptide. And this peptide will float away and go on to the next step, which we'll discuss in a minute. So it says here that it transfers it to a water molecule, meaning that what it does is the ribosome will catalyze a reaction by which there is hydrolysis of the polypeptide and that tRNA at the P side, and the end of that polypeptide is filled in with a water molecule. The peptide is free to go, and the ribosome senses that the peptide release, and the whole thing falls apart because now kind of everything is empty. The E side has an empty tRNA, the P side has an empty tRNA, and there's a release factor in the A side, no more peptide, so the ribosome can sense all of that, and it just falls apart. The ribosomal subunits will go on for the next round of translation. The small ribosomal subunit will have to find a bunch of initiation factors and a preloaded tRNA for methionine, and the large ribosomal subunit will hang out in the cytoplasm until it finds an mRNA molecule that has a small ribosomal subunit with the tRNA for methionine and initiation factors at an AUG. It will bind to it and start the whole process again. All right, almost there. I already kind of alluded to this. Is this a functional protein, or does it need anything else to do its job? I'll give you guys a minute to think about things you may have heard in previous courses or from the readings, but what else might one of these polypeptides need in order to become a functional protein, something that is useful to the cell? So if you did not peek into the next slide, what do you think can happen to this polypeptide or should happen to this polypeptide before it becomes a functional protein? Should get folded, okay. So I'm going to start writing it down. So it should get folded. What else? There's many right answers, by the way. So speak your minds. The protein or the polypeptide attached to something else? Attached as in it may start interacting with other things. I'll give you that. So we'll paraphrase your idea. So it needs to bind other things. I'm going to say bind other things. So I'm going to say bind others. Notice attached is a loaded word. It kind of implies some sort of covalent bonding of sorts. And that's not necessarily true. What else might a polypeptide need in order to do its job? All right. Okay, well, we'll go with these two ideas. So fold it and bind others. What folds a protein in cells? Whose job is that? Chaperones. Do you guys know what chaperones are? Proteins that help with folding. Yeah. That is very true. but it's also very special. It doesn't happen to all proteins. What folds most proteins out there? The protein itself. One thing that polypeptides do, if you guys remember your primary, secondary, tertiary, and quaternary structures of proteins. Yeah, coming back. So polypeptides have this ability to fold in space such that they acquire structures that make them energetically more stable, right? And for most proteins, or for most polypeptides, that's a self-regulated process. They do it themselves. Every once in a while, they will need a little help, and that's when the chaperones come in. Chaperones are gigantic macromolecular multi-protein complexes that can recognize proteins that show weird foldings, and what they do is, with a lot of energy, they stretch them out, and then they kind of guide the refolding in a way that is conducive to a functional final shape. Okay, so for folding, they really don't need much help, most of them. And then when it comes to binding others, that is true of those proteins that in order to become functional, they need several partners working together. Something that when you guys studied the biochemistry in proteins, you refer to as the quaternary structure. So primary structure is the chain of amino acids. Secondary structure is how that chain may either form alpha helices or beta sheets in space. Then tertiary structure is when those things start folding onto themselves further. And quaternary structures is when different polypeptide chains start interacting with each other. All of that is true, except that many times, or more often than not, polypeptides will need post-translational modifications, meaning chemical modifications that happen after translation, after these things have left the ribosome. Right? So there are chemical modifications that will alter the structure of those polypeptides in space, because by adding or taking away charges or neutralizing charges, they will force a movement in space of these polypeptide chains such that they can accommodate this new biochemical reality. So these chemical modifications can change the shape of proteins. There's protein trafficking to specific subcellular compartments. So a protein may be absolutely useless unless it is in the right place of a cell. And cells have protein trafficking mechanisms that will recognize certain we call them in technical jargon zip codes. There are specific amino acid sequences in the polypeptide itself that tell the cell this protein belongs in a peroxisome. This protein belongs in a mitochondrion. This protein belongs in a chloroplast. Check that out. Chaperone-induced refolding. That's one of the processes that a protein may need to go through. And then last but not least, proteolytic activation. That refers to sometimes a polypeptide won't be functional until something else comes in and chops off a little bit of it. When it does that, when that protease comes in and chops off a part of that polypeptide, now the rest will refold to adapt to that new reality, that new chemical reality for that polypeptide. And in refolding, will acquire a shape that makes it functional. We'll discuss all of this in more detail when we get there to gene expression regulation.